Okay. So brief. Done. Figuring out about the codon, the language from DNA to RNA to protein. And today we're going to find out how RNA is made into protein. So the specific process of translation. Before we do that, though, I just want to go over the questions that were assigned to you. All right, so you had 15, 16, and 17. That's what had to do with what we went over last week. And then today I'm kind of assigning to you 22, 23, and 24, which will have to do with what we're going over today. Okay. Remember, your next exam is coming up. It's on Monday, right?
Yeah.
Monday next week, yeah.
What chapter does it cover?
11, but 10, 11, 12. Okay. Exam. Translation, and then gene expression. Two on Wednesday. Okay. 4 codes, right? It's giving it to you 5' to 3'. So what are we looking at here? DNA or RNA? mRNA, right? Because there's U's in it. Right? Is it just kind of the, train of thought I want you to take when you start seeing these problems? So first, identify what kind of nucleic acid it is. And then it's asking, so what are the amino acids specified by the following mRNA sequences? So where we'd have to first, because this could be, when mRNA is transcribed, right, there are parts of the molecule that are not translated, untranslated regions, or UTRs. There's one on the 5' side, there's one on the 3' side. The only part that is actually translated is the area between the start codon and the stop codon. That is the only area that is going to actually have opposites. So all we have to do for these problems here is identify the first start codon, and then identify the first stop codon that comes after first start codon. What is the start codon in mRNA? AUG. So for the first problem there's your start codon. That's going to be the first amino acid, which is always methionine. Now, it's telling you it's a bacterial mRNA sequence. The first methionine, called the initiator methionine, is a modified version of methionine. It has a formal group attached to it. So you always want to indicate that as being F methionine, since it's the first one. If there are any more methionines in the molecule, it's just regular methionine. If this is initiator methionine, and then anything inside the transcript is going to be regular methionine. All right, so when you're scanning along here, the first stop codon that I see is UGA. There is no amino acid for UGA. Okay, so this is where the protein stops. And then all you have to do is go to the, what's that?
Isn't it UAA stuff?
AUG, yeah, it is. I missed that. That means the answer in the solution manual is wrong. AUG. Very good. Good eyes there. Yeah, so, okay, so this one's wrong. This one's all messed up. So this wouldn't even have, would this be a valid transcript? What do you think? No. I mean, you could just have a methionine F-met, I guess there, but that wouldn't work. All right, so what he's saying here, this is what you have in the solutions manual for this problem. And this is a stop codon. This is what the author, I think, indicated. But yeah, this is a stop codon right here. So it must have overlook that.
You don't look at it in groups of like three.
You do.
Once you found the starter, so if that's the case, then that is.
But if that's the AUG though, that's where it's got to start from. That's going to be the first. So that's your reading frame.
Right,
Like that's.
then wouldn't you read the next three U's?
It would read as A.
Oh, yeah, it read as AAA. I'm sorry. Yeah, it would read as AAA. Okay, sorry. I'm on spring break too, guys. Okay, so you got UUU, AAA, UUU, AAA. So there's only two different amino acids in there, phenylalanine and lysine, right? And then the stop codon has no amino acid. All right, you guys got picking up the details here. It's about that.
He supplied the chart.
Of course, yeah, and you'd be supplied the stop codon. But as you're working through these, they're more familiar to you, and it's good to know them because it'll make it easier to spot them. Like he was able to spot quickly, even though we didn't consider the reading frame there. AAA. Okay, so yeah, so your amino terminus, like that F methionine is on the amino term. All right, so in B, the only difference there is that the AUG is a little further down, right? So this is not translated, right? These are all the same, it looks like. They're alternating amino acids. And then the UGA is your other stop codon. Okay. Any questions? All right, with C, so yeah, nothing crazy here. Alanine. AUG is the first one. And so you just want to go through and read it carefully, right? Because if you started here, that would be wrong. You'd be missing up, it'd be out of frame. All right, the one below, it just has one amino acid in it. Good. Okay. All right. So here's this problem. We're getting a sequence. It's they're calling it the non-template material DNA. So what amino acid sequence will be encoded in the sequence? So the coding strand accepts substituting. So all you need to do, turn this in. So when you do that, you substitute. So the 5' end is still the 5' end, and so forth. So ATG becomes AUG, and then substitute T for U all the way down. And then you do what we did before. So this is 3. So there's no stop codon indicated here, but it could still get translated. It just might get stalled. Any questions? Okay. All right. They give you a short amino acid sequence. All right, tripeptide, methionine, tryptophan, and histidine. You have all the possible nucleotide sequences on this mRNA. So we have to look at each amino acid, and we put the all right, so we have methionine, trip, You guys see that? Okay, well, I can tell you that methionine has just one codon. And when you look at the chart, you'll see that. All right, methionine has one, tryptophan has one, and histidine has two. So there's two possible sequences that would come in sequence. Right, so just combine these together, then you combine these together. Those are your two sequences. All right, so the question that was asking, give all possible nucleotide sequences on the mRNA. It's also asking for the templates and the non. Right, so told you here how to, just like we did in the lab. Except that you have to take there. This is still going to be 5' end, right? So non-template is going to have that same polarity. And then the template strand is the reverse complement of that, right? So you just take the complement of it. Okay, so that's 15, 16, 17, and then for 23 and 24, going over today. And we'll go over those questions on Wednesday. All right, so the next mechanism that we're going to learn about is translation. Translation is the process of going from a messenger RNA third one. And the translation based on the triplet code within the messenger RNA. And then termination is when that stop codon is reached. There's a couple actions that happen that cause the whole thing to fall apart. Yeah,
A triplet.
the reading frame. Okay, so this kind of just reviews the polarity. Very important. There'll be a number of questions probably on the exam that have this. All right, so the way, instead of like having to memorize all this, I think probably the easiest way to do it is to just kind of learn this, right? And then be able to work forwards and backwards from that. Okay, so if you know that the start codon is always on the 5' end, and you know that this is going to be identical to the coding strand, you can determine anything from that, right? To avoid confusion. On the exam, you could always write it out. You can always write this out, basically at the beginning of the test and just refer to it. That ribosome is going to move towards the 3' end as it's building the protein. And as the protein is coming out of it, it's the amino terminus that's coming out first, and the carboxy end, which is being built. Okay. Okay, so one of the involved is tRNA. About that last time. It stands for transfer RNA. RNA molecule. So every time a tRNA So in order for it to get recycled, it enter the translational machine that's amino acid chain. The charging of tRNA molecules enzymes called aminoacyl tRNA synthases. And what's nice about these enzymes is there's only one per, it's not like there's one per all the different possible, for all the different possible, all the different possible tRNAs for codons. It's not just one per amino acid. So you'll have one aminoacyl tRNA that it's targeting one amino acid, but it could be targeting multiple different tRNAs because the degeneracy. If you have three different possibilities for one amino acid, you have three different molecules, as the anticodon is different in each of those molecules. This is just one per amino acid. So every tRNA on the 3' side has a CCA sequence. That's the terminal nucleotides on the 3' end. This over here is called the DHU arm. And it's involved in like the large ribosome when that tRNA comes into the complex. The charging reaction just involves putting the amino acid on either the 3' or the 2' carbon. It can be either one of that adenine nucleotide on the CCA 3' end. Okay, so this is the 3' end here. It can go here. All right, it is usually 3 that the amino acid is tagged on. All right, so don't confuse like this 3' here. This is the 3' carbon. Right? This is the 3' end of the A. So, all right, so once that amino acid is stuck to it, now it's charged. It's ready to, it can ribosome if needed. All right, so methionine that are generated, and that's because you have our codon. But then if any are needed, it's just regular plain old. Right, so we'll have two versions of tRNA methionine. So it's usually pretty easy to indicate referred to if it's the initiator or the plain old regular. It'll have the letter I in it. So I wouldn't get too wrapped up with this, just understanding that if you see the I, you know you're talking about initiator methionine. The molecule is different when you compare it to prokaryotes and eukaryotes. But it's just it's a different function that's added on to methionine when you're comparing prokaryotes. Okay, so where do amino acids bind on the tRNA?
C.
Yeah, the 3' end. DHU stands for deoxy base that's on that side of the, it's mentioned in the. Okay, so what are the different now? However, we're going to cover translation as compared to prokaryote. Obviously, the 30S, 50S is the large, the All right. Few initiation factors that are involved in translation. IF3 is the one that's involved in initiation. And then you need the mRNA. So the mRNA is going to have a start codon. Realized during the process of initiation, right? So these are all the plasm. See how it goes. So I say similar. All right. So what are the steps? The first step is that IF initiation factor will bind to the small ribosomal subunit. And what it's doing is it's preventing the large ribosomal subunit from binding to it. Because if that thing were to clamp down before, you wouldn't be able to proceed. Because what needs to happen is the mRNA needs to come in and stick to the small ribosomal subunit. Right? So IF3 is preventing the large unit from coming in. All right, so it can come in and bind because of the presence of the Schijn-Dalgaard. It's complementary to some of the RNA sequence that is found within the small ribosomal subunit. Remember, these ribosomes are made up of RNA and protein. Right? So that means that since RNA, they can have cap, and we're going to see that a little bit with the large subunit. All right, so after the small subunit is bound to the mRNA, there are some additional initiation factors that are utilized. So now we have to start thinking of the initiator methionine getting to the start codon. And that does happen first before the large. It's times when you learn this and you don't learn it that way. You learn it just the whole thing. Okay, so you need at this step GTP and also IF2. Okay, so you get a complex between the first initiator methionine with IF2 and GTP. And now that whole thing can bind to the start codon, right? The anticodon is complementary to the AUG. We have our modified methionine there, right? And the next step is you get all of these factors, F3, IF2, and IF1 associate. GTP gets hybridized, and now the large ribosomal subunit can come in and clamp down over this thing. Now the 70S initiation complex, right? So from the 30S and the 50S, now you have a 70S. Remember, you don't add it because it's Svedberg units. This makes up the initiation complex for prokaryotic initiation. So one thing about large ribosomal subunit is there's like three little kind of look like windows in it. We call them sites. You have the A site, P site, and the E site. They stand for aminoacyl, digital transferase, and exit. APE. Now, if you notice, when this thing clamps down, where is the tRNA, the initiator tRNA? It's kind of in the middle of the ribosome. It's taking over the P site of the large ribosome. Okay. After initiation, what is elongation? Okay, so elongation is going to use all three of and the P and the E. All right, so just a little note about eukaryotic initiation, more complex, is like in it. So how does small ribosome to the large ribosome are different, right? And the most difference, though, there's 12 initial significant, but in what happens is you get these proteins here that are binding to the cap, right? And you get proteins that bind to the tail as well. This whole mRNA loops around, it forms like a closed loop. And that closed loop helps kind of clamp all this thing together. So you don't see this in prokaryotes because there's no poly A tail, there's no cap. But in eukaryotes, you get the formation of these closed loops. Okay. Again, I just want to reiterate these are important. They space out. You have space between the poly. You have all that stuff. Actually, sorry, Kozak sequence is right here. Includes the AUG. Shine-Delgarno sequence is a little further. All right, during initiation, the small ribosomal subunit binds to which consensus sequence? We've already talked about TFR.
Shine?
AUG is a codon, so it's the Shine-Delgarno, right? That's the consensus sequence. If we were talking about eukaryotes, during initiation of translation in eukaryotes, small ribosomal subunit binds. How does it bind to the sequence? If I were to ask you that question. So if I were to ask you during the initiation of translation in bacteria in eukaryotes, how does the small ribosomal cap is what facilitates binding and prokaryotes and eukaryotes. So that Kozak sequence, remember, it's not important in binding of the small unit to the mRNA. It's important in finding the AUG. All right, so what do we need to do elongation of the protein? So we have the complex assembled. That large unit has those three sites, the exit, peptidyl, and the aminoacyl. There we have the total 70S complex, right? And then we bring in these three elongation factors, right, that are involved in the process of elongation. All right, so before I go through the process, I want to just show you a video of it because there's a lot of translocation terms. Because obviously with exit like that, tRNA patients. So there's a lot of movement going on. So seeing an animation can make it a little grasp. This is going to purpose of this.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site.
Right, so that's important. It's really just to see the mechanics of here. So the next
The
site
first
that is
tRNA
filled
occupies
is
the
the
P
P
site.
site.
The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid. The first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters.
You can see it.
These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
All right, so what is the process of elongation? All right, so remember, initiation complex assembled. Is in the P site. And at first we have the initiation factor with elongation. Elongation factor is abbreviated IF. These are abbreviated E. First one that's utilizes is EF2, and it has GTP attached to it. Okay, so you get EF2, GTP, and charged tRNA is corresponding to the in this case, it's CC, the anticodon GGG, proline. It's got EF with GTP, and with the enter the A site of the large ribosome. All right. So after it's in the A site, GTP EF2 utilization gets released. So after it's released, of the large ribosome in step 2. Remember, the ribosomes are RNA and protein, which means they can't have catalytic activity. The ability to form a peptide between the two adjacent comes from the large ribosomal subunit. It's not a specific enzyme that's doing it. It's the large ribosome that's actually doing it. That was a very significant discovery in biology because it kind of told us that, the RNA molecules early on, as life was forming on the planet, it was basically the RNA molecule because of the fact that they can have catalytic activity. All right, so you still have the A site occupied. You get the formation of a between the two amino acids. And now we have to do the translocation part. We have to shift everything. So to do that, the ribosome moves down the mRNA, it's the next codon. And the next elongation factor that's required is EFG. Okay, so that is all GTP. Right, when that gets hydrolyzed, you can get translocation. So it's hydrolysis of the EFG GTP complex causes the movement. So you can see here the tRNA is moved from P to E. And once it's in E, it's basically like ejected, goes right out. The main dancing that's occurring here is occurring between the A and the P sites. Once it gets here, it's out. All right, once you get hydrolysis of that GTP, you can get the translocation, everything two amino acids, and you have an. So this process can. It can come in and get put into the A site. Okay. Just some tricks on. If you're not seeing it at all, I would go back. It's moving through. Draw it out. To know this process in detail. All right, in elongation, the creation of peptide bonds between amino acids is catalyzed by what? So I really didn't pick up on this earlier, but none of those is exactly right.
RNA.
Yes, I'm sorry. Yeah, RNA. That is the right answer. That's a good answer. Not wrong with that answer. I was looking at B and C. Yes, RNA. Right. So it's the RNA component of the ribosome that is catalytic. It can form the peptide covalent bond between the amino acids. Good. All right. Translation ends when you reach a stop codon, UAA, UAG, or UGA. If there is tRNA, there is this thing called release factor that will come in and stick where the stop codon is. Okay. So what does that cause to happen? Well, basically the whole thing kind of comes apart. You get a release of the protein from the complex. You get a release of the tRNA that's in there, and you get a release of the mRNA. Things falls apart. All right, now there are a few different release factors. So really all you need to know is in bacteria, there's RF1, 2. I'm not going to get too crazy with this. I think really just knowing that F is in there and the stop. So that's all I've kind of put in. All right, so when you come across a stop codon, this is eukaryotic, prokaryotic, there's no tRNA, there's only release factor. It comes in to the A site. You do get a GTP, and then everything falls apart. That's it. All right, so this does the components that we've talked about today. You need to know all the initiation factors, difference between initiator methionine, the other methionines. Need to know all the elongation factors. Again, release factors 1, 2, and 3. You don't need to know the differences between those. Just any release factor is fine. All right, the last thing in this chapter covers something called polyribosomes. So what happens is when an mRNA gets to the cytoplasm, one transcript can be translated simultaneously in multiple ribosomal units. What's going to happen first, wherever that start codon is, you're going to get an initiation assembly event that occurs there. Right, so now you're building a peptide. So that was this one here, bound first on the 5' end of the mRNA. And it's moving down. But once that initiator codon comes out of the ribosomal complex, it can be translated again. So you get another ribosome that clamps down there at that start codon area, starts building a protein. And when that comes out, you get another one and another one. So when you look at these transcripts under a very powerful electron microscope, you can see presence of complexes using protein. So you get a lot of amp transcript. Now you can have when you're talking about protein that's made as well. That's what we're going to talk about in the next chapter is and the cells that they are expressed in. All right, the polyribosome, polypeptides associated with which ribosome longest? What do you think? Yeah, very good. 3'. Okay, those are your questions you should do before Wednesday. Don't forget to bring your genetically confirmed Wednesday.
Yes.
Sorry.
Yes. Yeah.